Thank you so much, Stephen, and thank you for inviting me to do this because I need to do it now and then just to, not totally forget how. This is kind of a presentation I wanted to do about 15 years ago, but the question about what makes a cat orange was only solved recently. So I'm going to be trying to keep up with my speakeasy as we go through the slides. So if you have questions, I will try to see them, but if I don't, then we can catch them at the end. So I'd like to start looking at the major pigments that are found in the placental mammals, and both the pigments come from the same branched pathway, melanin and or eumelanin, as it's sometimes called. That one is black or brown, and pheomelanin is yellow, orange, or reddish. And they both come from the amino acid tyrosine. And there is a branch point at dopamine where they separate. So melanin goes down one side, and pheomelanin goes down the other. So there's a sort of choice point between the two pigments. And the total color of the animal depends on the relative amounts of each one you have and the pattern that they're arranged in. For those of you who like structures, this is a simplified version of the pathway, but it shows some of the structure so that you can see tyrosine here and desmoquinone, which is at the branch point. Pheomelanin and melanin are not shown here because they are polymerization of these final bits of molecules. Yes, I like chemistry structures too. So there are two important regulators of the melanin pheomelanin pathway. One of them is MC1R, which is the melanocortin 1 receptor protein, and that is activated by MSH, the melanocyte stimulating hormone. The gene locus for MC1R is usually called E for extension, which basically means how far does the pigment go. The other protein is ACIP, the agouti signaling protein. And ACIP also binds to MC1R and competes with MSH to produce different kinds of pigment patterns, stripes, spots, dark patches in various parts of the animal's body. They make very interesting patterns in German Shepherds, for example. If ACIP is inactive, then the animal is all black, and that is a gene at the A locus. And I think I'm getting behind. Hold on, I got to catch up here. Oh my, I got way behind. Okay. So when most of the pigment is pheomelanin, you get things like chestnut horses or golden retrievers or red-haired humans or tigers, yellow tigers or golden tigers, and yellow mice. Now, this is a diagram of the MC1R protein. It's a transmembrane protein, so it crosses through the membrane. It goes back and forth across the membrane several times. The MSH binds up here on the outside of the cell, and then the activating parts of the protein are down on the inside of the cell. And this is active in melanocytes, which are pigment producing cells. Okay, so just a couple of examples of color variations. Here's some examples of the effect of the ACIP and the extension genes on horse color. If you have an inactive ACIP and you have a dominant gene for MC1R, then the horse is black. The wild type, I guess, is this bay horse here. And if you have both genes active, then you get a horse that looks kind of like this. Its body is sort of brown. The mane and the tail are black. And it also tends to be dark on the ends of the legs. And in chestnut horses, got both dark and light chestnuts here. They're a little bit different, but they both have the recessive allele at the extension locus. And they don't have the intense black coloration that you see in even in bay horses. And then in dogs, that same combination of genes gives you black Labradors if you have the dominant alleles at both locus, and yellow Labradors if you have the double little e genotype. Um, Interestingly, the skin in yellow labs is still pigmented. If you look at the base of a yellow lab's coat, you can see pigment in the skin, especially around the face and the nose. Okay, so that pattern of color variation with interaction between MC1R and ACIP occurs in horses. I mean, it's been demonstrated in all of these animals: horses, dogs, big cats like lions and tigers, mice, humans, and bears, and most other eutherian mammals have these color combinations. Yeah, like black squirrels. Um, would be dominant for the A locus, but not dominant for the E locus, but not for the A locus. So the black squirrel would get you that. So most mammals then use that pattern for pigmentation. So what about domestic cats? So this is a nice picture. Oh darn, I lost track of my speak easy is not that easy. Let me catch up with myself. Okay, so I'll try to do better. All right, so this is a nice cat named Dexter. And there, as I mentioned before, there's a whole website with pictures, lots of pictures of orange cats. And if you want to look at it, I've got the links there. So cats must have a mutated MC1R. So where is the MC1R gene in these animals? In humans, it's on chromosome 16. In dogs, it's on chromosome 5. Horses, it's on chromosome A3. In mice, it's on chromosome 8. And in both big cats, like lions, and in domestic cats, it's on chromosome E2. So this is a problem. And here is the catch. Different chromosomes are numbered differently depending on when the chromosomes were identified and who named them and so forth. There's not one single method for numbering the chromosomes. All right. So what we've known for more than 65 years is that the cat orange gene is not on chromosome 2, it's on the X chromosome. It's sex-linked. And in 1961, Mary Lyon proposed that in female mammals, one chromosome must be inactivated in females to maintain balanced gene activity in males and females, because females have two X's and males have only one. And one of the reasons she thought that was that she had noticed that nearly all calico cats are female. And the females express, if you've got a heterozygous female with one orange gene and one non-orange gene, then the orange gene will be expressed in some patches of tissues and only the non-orange gene in others. In fact, you can't actually tell which of the two genes is dominant because by looking at the cats, because one X chromosome is always inactivated. So only the genes on one of the X chromosomes are going to be expressed in a particular patch of tissue. So the MC1R gene is not located on the X chromosome. So how do these cats get yellow? Sorry, catching up again. I'm really not good at this. Okay. Well, well, I see something in the one thing I didn't mention. Uh, Humans also, all mammals inactivate one X chromosome in females. All the placental mammals do that, including humans. And we don't get calico humans because MC1R is not on the X chromosome. So you don't get selective inactivation in that sense. Okay, so where on the X chromosome is the orange gene? And what genes are at that location? And how might it be connected to MC1R? And this is just a brief rabbit hole. I'm not going to stick with this too long. If we go back to the previous slide, which I will do now, you'll notice one thing, and that is you only see striping in the orange patches. And these cats, these two particular cats, um, the black patches are not striped and the orange patches are striped. And this is due to differences in the activity of that ACIP protein. Okay, but um, in cats with an active ACIP, you see striping across both the black and the orange regions, and the striping is continuous across the color line. So that's another interesting question, which we're not going to go into anymore here, but might come back at a different time to talk about that. Okay, so back to where's the orange gene? In 2009, there was a group at the National Cancer Institute, headed up by Anne Schmidt-Kunzel. Sorry, I didn't get the out in there. Um, and they looked at which set of microsatellite markers. Microsatellites are very short tandem repeated sequences like CG, 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 something like that. And they're about 3 megabases, 3 million bases apart on the X chromosome. And they found that in orange cats, there was a region on the X chromosome that was consistently that had the same group of microsatellite markers and was consistently associated with the orange trait in male cats. So a lot of recombination goes on between the two X chromosomes. So they're looking for a region of the chromosome that is always retained in the orange males. And the most likely candidate was between 106 megabases and 116 megabases on the X chromosome. And there was a 9.6 megabase stretch in there. And this is kind of a picture of that. So this just these things here are the satellite markers, and these are the ones that are not showing crossing over between themselves and the rest of the chromosome. That is, you get crossover in this region and you don't get an orange cat. So this is the so it's in here somewhere. It's in here somewhere. All right, now, so they found that location. They looked in that region of the X chromosome because the X chromosome had been pretty well sequenced in humans. And so they thought, well, we can just look in there and we can find the gene. But they did not find anything in that neighborhood that was known to be associated with pigment formation. So it was just everything came screeching to a halt at that point. They couldn't figure it out. Now, red hair selection is like chestnut horses. So it's not the same in humans. Humans are like everything else, but only cats, only domestic cats seem to exist so far. There may be other animals that aren't known yet to do it, but so far these are the only ones that do it this way. So over the next 16 years, that candidate region was further refined until two independent research teams, one of them at Stanford, and the other one at Kyushu University in Japan, both published articles in the same issue. They were working on a problem simultaneously and they came to the same conclusion. And they published their work together in the same journal in the same month. And both of them had identified the gene locus was a gene called ARH-GAP6, the Rho GTPase activating protein. Don't worry about that, what that does right now. Because the deletion wasn't in the active part or the transcribed part of the gene or the translated part of the gene. It was in an intron, and introns in eukaryotes you have alternated exons and introns, so the gene isn't continuous through its structure. Its translated parts, its protein coding parts are kind of scattered throughout the gene. So the introns, don't contribute to the protein. So this deletion wasn't even in the active part of the gene. It was in an intron um, at the beginning of, of the transcribed part of the gene. So here you have the whole X chromosome, and in this orange region is the region that had first been identified with with orange coloring cats, and then the ARH-GAP6 is in this region here, and then that deletion is found here. Now, here are three different transcripts of the ARH-GAP36 gene. And you notice that they're different in that they start at a different exon. So exon 1 is over here in this version of the gene. In this version of the gene, it's here. And in this version of the gene, it's here. It's not that the genes are different. It's that transcription can start at different locations. So there are several different transcriptional start sites in this gene. And that deletion is in the big intron that's at the very beginning of one of those transcripts. Uh, Down here, it shows what the transcriptional uh, picture is in different kinds of cats. In orange cats, there's a big gap in the transcription, except for itty thing right here, which I think is interesting. And in regular cats, in non-orange cats, you have transcription all the way across that region. And in calico cats, which have one orange gene and one black one non-orange gene, you just get half the amount of transcription because only one of the two X chromosomes is producing those transcripts, the one that doesn't have the orange gene. Okay, so ARSCAP6 has these three possible transcriptional start sites. The deletion is at 5.1 kilobase, 1,000 bases, located in a large intron between the first two transcriptional start sites. And since it's in an intron, it doesn't affect the function of the ARHGAP6 protein at all. And in both the Stanford and the Kyushu studies, all the orange cats they collected from around the neighborhood of the universities and other cats that they found out about from other parts of the world had that same deletion. So there seems to be just that one orange variant in cats, known so far at least, and that probably had a single mutagenic origin at some time in the history of the domestic cat. So we've got that strange orange variant, and it is only found in orange cats. So what's in there? Cat. Speaking of cats, cat is rocking my screen at the moment. Okay. Um, so if you compare that the content of that deleted region in several different species, you find several things. One, there's a very highly conserved 436 base sequence found in all species. Um, you also find in cats that the region contains, and also other animals, contains multiple possible regulatory elements that might either enhance or repress transcription of other genes. So there are genes in there that are known to be regulators. And the Kyushu group looked at the expression of these 13 genes in the O region and found that the ARHGAP6 was always expressed in orange cat tissues, but the other genes in the region were variably expressed. So the ARHGAP36 gene is not only not affected, not inactivated by whatever is in there, but it is very highly expressed in orange cats. Okay, so how is this affecting melanogenesis? We know that the orange gene has to be, the action of the orange gene, that is what it's doing, has to be downstream of MC1R activity. The MC1R activated genes important in enhancing the melanin pathway. These are the ones that make the melanin dominate in animals. Are protein kinase A, PKA, pre-melanosome protein, PMEL, a microphthalmia associated transcription factor, that's a jawbreaker, MITF, and TRYP1, the tyrosine related protein. That one also has an interesting story, but we won't talk about it right now. And dopamine tautomerase. So reduction of these proteins would favor pheomelanin synthesis and thus produce an orange animal. So the activity of various genes related to that, the melanin pheomelanin switch, was looked at in cat cell cultures. So they took, bits of tissue from, I think, the ears of the cats, and then they put them into cell culture and they examined what was going on in the genes in the cell cultures. Okay, so what did they find? Well, oh, wait, quick commercial. Um, this just shows what's going on in the melanin producing branch of the pathway, and the different genes that I mentioned are all here. And this one is particularly interesting because it triggers the pathway that involves all of the others. Okay, so if you look at the increase in ARHGAP6 expression, it's associated with the reduction in the expression of several of these genes. All of these genes become less active. And those are all genes important for eumelanin, the choice of eumelanin in the biosynthetic pathway. So the fact that several of those genes are inactivated suggests that what they have in common might be the inactivation of PKA. So they looked at actually human melanoma genes, human melanoma cells that have been transformed with the cat ARH-GAP636 gene and then activated with the melanocyte stimulating hormone. And it found that those cells showed a reduced expression of the catalytic subunit for PKA. That is the subunit that allows the enzyme to be active. Okay, so this is just the pigmentate, just the stain marker that they were using, and this is the ARH-GAP6 that was tagged with that stain marker so they could see where the PKA was being produced, and they found there wasn't much of it um, in the presence of ARH-GAP36. Okay, so to sum it all up and to catch up, let me show up. Okay, so we know that the real culprit in orange cats is that PKA gene. So the MC1R gene is normal in orange cats, and it's located on chromosome E2, just like it is in the big cats. Little cats are just like the big cats. On the X chromosome, you have ARH-GAP36, which is consistently expressed in orange cat tissues. Loss of a regulatory gene in the deleted region in an ARH-GAP6 intron increases the expression of ARH-GAP6 in orange cat tissues. The ARH-GAP6 reduces the activity of the PKA catalytic subunit, which is necessary for the expression of that whole cluster of genes that is downstream from PKA in the eumelanin biosynthesis pathway. And removing the melanin pathway then favors the pheomelanin pathway, which is the red pigment, and that's how you get yellow cats. Okay. And if you want to read any of these, here's some of the references that are cited in the pictures. And these last two here are the report of what the yellow gene actually is. So if you want to read those, you can. Okay, so questions. Oh, okay, let me start with Phil here. Yes, all orange cats have some kind of mutation. Now, the other orange cats, humans and mice and dogs and horses, have different mutations in MC1R. They're all mutations in MC1R, but they're not all the same mutation. So there's a different one in humans than there is in horses, than there is in the big cats and so forth. Okay, so those aren't all the same, but they are all mutations in the same protein. And let's see. Oh, well, some breeds of cats, and I can't, I don't actually know that much about a bunch of cat breeds, but some breeds of cats are not allowed to have orange offspring. That is, in order to, sell that breed of cat, it has to not have an orange gene. So, but those have been removed by selective breeding from, cat breeders. No, that orange mutation in cats is all the same orange mutation in little cats, but it's not the same as the orange mutation in everybody else. So there was an individual mutation in the ancestor of domestic cats that popped up at some point, and all of those seem to be the same mutation. So that would be probably an individual mutation. Um, because there aren't like variants of it. There's that one deletion, um, which is found in all orange cats. Yeah, the white spotting is due to a different gene, which is also interesting, but I'm not going down that rabbit hole. So you have a Maine Coon that's orange. Is it a male? Because of because the female cats have to have two X chromosomes that have the mutation, there are fewer female orange cats than there are male orange cats. So if you see an orange cat, it's maybe 80% likely to be male and 20% likely, if possible, to be female. Um, I haven't seen a, I haven't seen any information on that, but somebody may have figured out when it first appeared. But that's a good question about when was it first seen? When did the first orange cat turn up? And um, it's certainly very possible that it was before written history. Yeah. Yes, you got two kinds of cats that can have a one orange gene and one not orange gene. Some of them are tortoise shells, and they just have different patches of orange fur and gray or black fur. If you only have the orange pigment, then you've got two copies of the mutant gene. If you're a calico, you have one copy of the mutant gene, and you also have a gene for spotting. So that's the difference between calicos and tortoiseshells. Calicos have the spotting gene, and tortoiseshells don't. Well, the difference between an orange cat and a calico cat is the orange cat has two of the orange genes, and the calico cat only has one. Since they're on the X chromosomes, that one is expressed only in the tissues that have the X chromosome with the orange gene activated. If you've got the X chromosome that has the non-orange gene, then that patch of fur will be black. Is that making sense? The white fur is due to the spotting gene, and that's an interesting gene because the dominance in that case is intermediate. If you've got a very white cat with a big white surface, you'll notice that the pigment tends to be concentrated on the dorsal side of the cat, on the head and the back and the tail, and maybe the rest of the cat will be white. Um, a white cat with gray markings. Okay, so a gray and white cat. Oh, okay. So do you have a gray and white cat with a gray patch in the shape of a heart? Is it on the back? Okay, well, what you got there is color dilution. Um, whether an animal like a horse, for example, is gray or black depends on some other genes that influence the pigmentation, and one of those is a color dilution gene, which reduces the melanocyte activity. So if you've got the orange gene and the cat is sort of cream colored, you've got the black gene and you've got a gray cat. So that's a combination of having a not orange gene and the deletion gene. So the white is due to the spotting. And if you have extensive spotting, then the only gray parts of the cat might be it's part of its head and its back and its tail. Oh, you got the date on it. Okay. Oh, unknown. Okay. Early in the cat domestication process, roughly 900 years ago or earlier. Ginger cats appear in historical art dating back to the 12th century. So we know it's been around for at least 900 years. That's using cats in paintings as a historical marker is a good idea. Yeah, tuxedo cat has just a little activity at that spotting gene. Well, they might evolve faster than humans because they reproduce. They get to reproductive age faster than humans do. You know, when a cat gets to be a year old, it's pretty close to its reproductive age. In fact, it's probably younger than that. And humans can't reproduce until a little bit later. So, um, and domestic cats have probably been around less long than humans have. So maybe they might be able to evolve faster, but they may not, they've probably not been around as long as we have. And there are some fascinating patterns in cats. One of the sources I was reading said that, you know, you could teach genetics entirely just by using cats as examples. They do also have shorter lives, but it's the span to reproductive age that makes that turnover faster. How many offspring? What do you mean? An average male cat has well, that depends on the female. Male cats do get around, and of course, they they can reproduce a lot faster than females because they don't have to go through pregnancy before they can, you know, get the next one. Meow. There are some calico males. They are very rare. And you might be able to figure out that they have two X chromosomes, um, and a Y. They have to have a Y chromosome to be male, but they also have two X chromosomes. So there are rare male calico cats. They usually are not reproductively active, um, probably because of the odd number of X chromosomes. Okay, well, I'm glad that I finished this as soon as I did because that means I can go to Ari's concert.